It's my pleasure to welcome you everyone now to this session. And my name is Thomas Berendonk. I'm from the Institute of Hydrobiology, TU Dresden, but I'm not important. Much more important is the speaker today, and I feel very honored that I can introduce him. It's Zhang Tung. And I think probably all of you know his name. I mean, if not, you really need to do some improvement on your reading. He has an H index of 110, so you should know that name. He's a chair of Professor of Environmental Engineering of the Microbiome Engineering and Biotechnology Laboratory, that's a really long name, at the University of Hong Kong, and he's also a professor of the School of Public Health. And I think most of you know his work, which is very encompassing, and most of us know him because of his bioinformatics skills. And with that, I really would like you to start, and again, welcome.
Thank you, Thomas. Thank you for a kind introduction. And also, first of all, I would like to thank for the invitation from the Scientific Committee of EDAR7. So give me this opportunity to come here to share with you some recent studies and also some thinking about how we need to move forward from now. As you can see on the screen, I put my cover slide back to 12 years ago when I attended the first workshop organized by professor at the top. Although I can put my slide 12 years ago here, but I do not have a time machine. We cannot come back to 12 years ago. Today, when I stand here, I want to share with you some about good memories. Now, one day, back to 12 years ago, I got an email from people who I don't know. Then I look at the internet. I found this nice picture with very natural smile and with a very simple model of risk assessment. Then I take the adventure and decide to come and to join this first workshop organized by Professor at the top, organized by Ed. I think Ed is, I saw Ed walking in the room, yeah. Oh, you are here, you are there. Oh. So as you can see here, the name of the first ADA, actually it's entitled as the Antimicrobial Resistance in the Environment, Assessing and Managing Effects of Anthropogenic Activities. And that's the original name of this conference. And at that time, there are about 40 people. Yeah. And my adventure to the first ADA became a happy and memorable experience. I stayed in this luxury and historical hotel, and also meet with a group of nice, kind, and smart people to talk on the different topics which we have a common interest, and have the excited talk and also fruitful discussions on the topics which you see here. That is about 8 topics. We have group discussion, and you can see on the screen it is about the exposure, the impact, and also some evidence, and also some research approach, and finally it's about the risk assessment and modeling. Yeah. When I look at this list of these questions we try to answer and discuss at that time, I think still keep open. Yeah. Although, like, I guess there could be thousands of related papers published during the past 12 years, but the question is still there, and we still need to work harder on these kind of questions and try to find some solution. Although some solutions, I think, is already on the way. So today I'd like to share with you, I still work on the first topic, which astounded to me. It's about exposure. It's about exposure, how we assess exposure. Now today the topic which I will share with you is the standardized metagenomic absolute quantification using the long-range sequencing for the environmental surveillance of ARGs. As you heard from Thomas, my home Department of Civil Engineering, and since my work is more and more related to the public health, so a year ago, I was appointed as the professor by courtesy in School of Public Health in our university. And I think no necessary to waste your time on this One Health framework. For the attendant of this EDAR7, basically we all know about this concept and the framework and its significance. And for me, I think it's just a historical lesson and examples which many people already take it for granted. My background is on the environment science and engineering, and water treatment, wastewater treatment is an important part of that. And if we look back in history, back to 200 years ago, the British engineer already started to build up the sewer system and try to cut off the transmission chain from the human fecal waste and exposure to the human being. And then about 100 years ago, we have the wastewater treatment process using active sludge in Germany, and also the largely application of disinfection of our drinking water, which together with the sewer system, then they save millions of lives. The water treatment saved millions of lives. And in the past century, the lifespan of human being averagely, the whole world, increased from 38 to 68, about 30 years increase. As you may have heard from Gary at the opening speech, for Canadian it's about 20 years due to the invention of antibiotics, but for the whole world it's about 30 years. I think when people look at what mainly contributed to the increase of lifespan of human being, and antibiotic definitely is one of them, and the vaccination, and also water treatment, which I consider that is the historical lesson and example of the One Health concept and framework. And also another important things I think we may agree here that if we do not spend money at the source control, we'll spend more money in the hospital or the community. And as an engineer with the engineering training background, and usually the solution at upstream is more cheaper, cost effective than the solutions at the downstream. But it's very difficult, not that easy, to convince people until something bigger happened. And let me share with you a story, ancient story in the Chinese traditional classics. It's about Bian Che and his brothers. And Bian Che is a very famous doctor in ancient China, back to more than 2,000 years ago. And there's a lot of legend about him in Chinese classic text. And one story is about his and his brothers. One day the king asked him, "You and your two brothers are all skilled in medicine, and who is the best?" That's a challenging question, right? And Bian Che answered that, "My eldest brother is the best. The next is my second brother, and the last one is myself." Then the king is very surprised and say, "Then how is that you are the most famous one and well known to the people?" And Bian Che said that, my eldest brothers just treat the disease when it's just at the beginning. And most people don't know that yet. And my second brother treats disease when there's some small symptom, and very early stage, then it's only known by the neighborhood. And myself, when the things getting worse, so I take very heavy medicine and even some surgical actions. Then the people see that how I do that with hard work and great effort. Then people think that I'm good. So this is a story. Just take it as story. It may not be the real history, but the lesson is real and true, and it tells us that prevention is better than control, which in turn is better than remedy and after the event. I think it seems applicable to the AMR issues. If we ask Binche and old brothers where we should take action, I think the answer could be we need to take action in the environment before it affects human being and animal. And the roadmap to tackle the AMR in the environment, I think for most of us, it's straightforward. Although it's still challenging, but it's straightforward. It's not that blurred, it's clear. First of all, to identify the problem. As you may heard from Amy, whether the environmental AMR is a pollution issue or not. Back to 12 years ago or even earlier, still some argument. But now, I think it's quite straightforward to most of people. It is an environment pollution, not other. And also, the second, what we need to do is to survey the exposure level. Both the resistant bacteria and the resistant genes, maybe together with the chemical pollutants of the antibiotic itself and some other drugs which may cause co-selection. And with organized, international, organized, systematic and joint effort using the standard quantitative measures. Because this is global pollutants, just like the carbon dioxide, the greenhouse gas, and also some other like mercury. These are the POPs. These are the global pollutants. AMR also is a global pollutant. You solve problem in one area, doesn't solve the problem in another. So we should take that as a global pollutant. And also another important thing is to understand the mechanism of development of resistance in the environment, including the multiple selection pressure, the available concentrations in the microenvironment instead of in the bulk water, and also HGT rate, co-selection, etc. And also most important is assess the risk of antibiotic-resistant bacteria and genes of different exposure level. Need that quantitative information and comparable data set using standardized method. And after that, hopefully, we may establish the scientific criteria which does not cause impact or develop a regulatory standard for the priority for the priority ARGs with high risk. And the last one, definitely not the least one, is to implement the mitigation measures at those hotspots and important points. For example, the pharmaceutical wastewater and also the hospital wastewater, animal farm. Those are the hotspots for sure. Even with no necessary to collect more information. We know that by the education guess, we know that it's a hotspot. No need, although we still can collect more information, evidence, but based on the education guess, we know that it's a hotspot. And technology is already available to build up the barrier, just like the border check. We do not let the bacteria to travel free from those pharmaceutical wastewater to the environment. And do not give them the visa. And we should give the visa to the people attend the international conference, but don't give the visa to the bacteria from one compartment to another compartment. So the technology is already there. I think membrane and other technology is available. And regarding the risk of antibiotic-resistant bacteria and resistant genes, And basically, there are two types of risk. The first one is about the medical therapy risk. When we applied the antibiotics in the hospital, in animal farm, it doesn't work. But that is not that related to the scope of this conference. And the second risk, the generation of the antibiotic-resistant bacteria and resistance genes, and also enrichment spread of them in the environment, which also finally cause direct threat to the human and animal. That's scope of this conference. And usually we talk about the resistance using two terms, ARB and ARG. And in the environment, the live cell, they can carry the resistant genes, and they are viable cell carrier of resistant genes. But the dead cell, and even the DNA in the extracellular polymeric substance also can be counted as the kind of source of ARGs in the environment. So they all contribute to the antibiotic resistant level in the environment. And also both the cultured and the uncultured bacteria may carry the ARGs. So in the environment study, usually we just use the molecular method, which can measure the antibiotic resistant genes to indicate the ARB without or with cultivation of the bacteria. First of all, when we do the metagenome analysis, we need to have the database and the metagenome pipeline. And here just a short promotion about our pipeline. And some of you are using that, and I hope they can help you. And if you have some problem, just give us feedback. We can further improve that. And basically, we try to develop it into one for all. Basically, you get this one pipeline. You can get all the different analysis for the antibiotic-resistant genes in the environment. And very important things in the most updated version for this pipeline is that we propose to use a universal unit to express the level, the relative abundance level of resistant genes in the environment. It's a copy per cell. And I think that is a biological meaningful unit, so better compare with some other unit. So for detail, I think you can look at these publications a year ago. And you can see on this slide, it shows the relative abundance of antibiotic-resistant genes from the different type of environment. And it can be 3 magnitudes difference, more than that. And with the new development of the technology, especially the long-read sequencing, we have new tool, or you say new toy, to play in the measurement of exposure level. And if you look at this news feature on Nature Measured, they select measured of the year. It's long-read sequencing in the year of 2023. And starting from 2014, my lab using this technology to work on the antibiotic-resistant genes. And recently, our lab is recognized as a demonstration laboratory by ONT company. If there's some conflict of interest, I need to claim here, just to add kind of clarification about possible conflict of interest. But for your information, I didn't get any discount from them, okay? So if you look at the comparison of the short and long range, there are a lot. But the most important thing is the long range is really long. And you can see it can get up to 100, averagely, can up to 100 kb. And it gives us a lot of advantage to identify the, the host and also the mobile genetic element. And this table I summarized from the answer of ChatGPT. I asked ChatGPT, he gave me a table, but something is not that correct, so I modify it a little bit. And back to 2019, we have this publication that used the first try used nanopore sequencing to have the host tracking of ARGs with in the wastewater treatment plant using nanopore. And as you can see here, we can know the host and also know the type of resistant genes carried by a host. And also, this should that the red color should that the antibiotic resistant genes on the plasmid. So most of the resistant genes is actually carried by the plasmid. And recently, we developed a new pipeline to be applicable to the long-read sequencing and use the marker gene, and we can use that to calculate the number of the cell covered by a data set which you obtain using long-read sequencing. And for detail, I think I do not have time to go straight. And this already is on the website, on the internet at the bioarchive, and the manuscript on the revision. Hopefully, it can accept it in the coming months. And another thing I think we should keep in mind, especially when we have more and more samples, could be 100, could be 1,000 samples, and when we do the extraction, when we do the sequencing in different batches, then there are some variabilities, from batch to batch. How we can watch that and also maybe do some correction of that. So suggesting that we can prepare the so-called environmental reference material, ERM. So the simple idea is that you just take active sludge. For us, it's easy to take active sludge. Of course, if you work on soil, we can have another approach. If you take active sludge in a large volume, and then you split into different, very small volume, and it could be hundreds subsample, and then you take a few of them for the sequencing, and you look at reproducibility. And if the reproducibility is good, that means that they are homogenized, then you can use it later with other samples as the kind of benchmark for comparison to see whether there could be some bias or some strange things happened in your sequencing process or in your DNA extraction process. So that's one of the application, and you can refer to the reference we have published that. And another important thing is how we can do the quantification, absolute quantification. And if we look at the chemical analysis, I think we already know for the chemical analysis, we can add some spike in reference compound together with others to be analyzed. And the same idea, you can add some species, some bacterial cell, which not exist in your sample. Here, the work done by my PhD students is using two isolate from the ocean, and usually do not have that much these two species in your environment sample. Then the low relative abundance will not affect your analytical result. So you add it in, and then you look at what could be the recovery rate for that spike in. Use gram-positive, gram-negative, you can represent the two groups. Of course, if you have multiple, you can represent more, and have better benchmark. And as you can see here, the results show that, the results show that after correction, you just spike in, the reproducibility getting higher and more comparable for the different batches using the different DNA extraction kit or different DNA extraction method. And using the absolute quantification method, we can get the information about ARG carried by different genera related to the escape gene and also E. coli and vibrio pathogens. So basically, you can get absolute quantification about the different pathogens and also the absolute quantification of the ARGs carried by different genera of these different pathogens. Of course, talking about the pathogen, we should go to the species level or even strain level. I think currently it's possible to do that, and with more and more accumulated genome, that will be more more easier to do that. And also, using absolute quantification result, you can evaluate the removal efficiency of antibiotic resistant genes in different wastewater treatment plants. Here, just three of them. The red one is the membrane filtration wastewater treatment plant with the membrane biological reactor. So you can see the removal efficiency is much higher than the second one, the blue one. The third one, YL, is the biological treatment process, secondary treatment. And the blue one is just the chemically enhanced primary treatment. So you can see the treatment process is really different, and you can choose this kind of information. And another important issues when we evaluate the risk of antibiotic resistant genes or pathogens in the environment, that whether the bacteria they are live or dead. And actually, there's a very basic approach to do that. And for us, we use PMA. And the PMA can bind with DNA of that cell or naked DNA, so they cannot be amplified. They cannot be sequenced by nanopore. And you may say, how we can prove that? That is quite simple and easy. You have bacteria A and bacteria B. You keep bacteria A alive, bacteria B died. You kill that, and they mix them together. Then after PMA treatment, you cannot sequence bacteria B. And the next round, you switch them. You keep bacteria B alive, and then you cannot sequence bacteria A after PMA treatment. But before PMA treatment, you can sequence both of them. So that is already be proved. And we have some trials studies for the beach water in Hong Kong. And as you can see here, they show the difference of the antibiotic resistant genes, the subtype in the total cells and also in the live cells. And the abundance also shows some difference. And currently, we are using the established method for some systematic study in Hong Kong. A big project lasts for 5 years. Next year will be the last year. And we want to have the systematic surveillance and also risk assessment to identify the critical point for control. And I think this kind of elements, like the environmental reference materials, cellular spike-in, and the viable cell count are the key components in the analysis of antibiotic resistant genes and also microorganisms. Basically, in general, all the microorganisms can be analyzed in the same way. And finally, the advances on this aspect and effort on this aspect will lead to an emerging and also important new discipline, which is environmental analytical microbiology. And just like the environmental analytical chemistry, which we have now, and I think that later on we definitely will have this new disciplines to work on. And for the new disciplines and also for the fight against with antibiotic resistant genes, I think it's a long journey and can last for decades. So we definitely need new blood. And over the years, my lab trained the PhD students. Here, as you can see on this slide, around about 30 PhD students. And now most of them, they are working in the top universities of mainland China, and some of them working in Hong Kong, the university in Hong Kong, and also in Singapore. So they are the new blood and work on these directions. And hopefully, I think you can have more interaction with them during this conference or maybe some other events. And finally, I would like to thank the hardworking students and team, and also the financial support from different grants. And also, we have a few posters, just a quick promotion of the posters. You can look at our posters 13, 35, 73, and 101, and 114. Thank you. Thank you for your attention.
Hello, Ishi Keenum from Michigan Tech University. Really interesting talk. I had two questions. So one is, what's the detection limit that you're getting with your new kind of standardized sequencing? How many gene how low can you get?
Okay, that really depends on the original concentration of your sample. For drinking water, for the influent of wastewater treatment plant, the detection limit is different. Yeah, so they're not that complicated, but also not simple calculations, which I may not have time to share with you, but that depends on the concentration of the sample. Yeah.
Okay, and then my other question is, if we get everyone in the world to do the same method or the standardized method that you proposed, what suggestions do you have for standardized reporting and then compiling of metadata so that we could actually start to compile some of this data into like a global data set?
You mean if everybody do the same in the same way? Yeah.
Yeah. So is there like reporting that we need to make sure we're all doing so that we are able to compare our samples?
I think that needs an international organization or institution to organize that effort. Yeah. But that could be one way to have this homogenized and standardized method, and for the data to be compared, can be compared with each other and also shared with each other. So hopefully later on there could be like UNEP or the other international organization may organize this kind of effort. I know some international foundation like Bill Gates Foundation and also Rockefeller Foundation, they have some interest to move this forward. Hopefully they can pump some money in and then move this kind of forward. Thank you.
Hi, thank you for the great talk. I'm Aline, I'm a postdoc at Auburn University in Alabama. And I have my question for you is, how do you deal with the inherent compositionality of the sequencing data, especially like in low reads in SARG and the ARGs OAP, which I have used in my research, great tool. But it's something I've always been curious about. How do you deal with the compositionality of the data when getting your absolute abundances?
So for that question, I hope I understand your question correctly. I think first of all, you get the number of the cell by counting the essential single copy genes and normalize against the gene length. And the second, you get rates related to the antibody resistant genes and then normalize to the length of the specific type of resistant genes. And then you got a copy of the resistant genes. Then with these two numbers, you get a copy per cell.
Oh, I see. It's because, so for example, with Illumina, the output of reads is not linearly 100% correlated with the amount of cells that you put in. Mm-hmm. So like, how do you correct for that? How are you able to make that prediction of how many cells were like?
You need to do some quality filtration. For the data, you need to, although nowadays not that necessary to do the quality control of the data since the accuracy is already very high. But it still depends on the data set which you get. Some make not exactly uniform length. Some may not have the uniform quality. So still need to do some quality control of the data set.
Okay, thank you.
Hopefully this can answer your question. Thank you.
Thanks a lot for the opportunity to speak to the organizing committee, and thanks to you all for your attendance and your attention today. My name is Peter Stojas. I'm a senior research associate at Biozone at the University of Toronto. And today I'll be discussing my ongoing work on trying to identify the origins of particular type of resistance gene, ARG, sulfonamide resistance genes, and also trying to understand the complexity and the size of the gene family overall. So sulfonamides, also known as sulfas, are actually the first class of antimicrobial agent, the first fully synthetic antimicrobial class. They were developed in the 1920s and 1930s. And these, there's about 15 of them that are clinically approved. They're used for a variety of human conditions as well as in veterinary medicine. And they target the folate pathway, which is an essential pathway in all bacteria, the de novo tetrafolate synthesis pathway. There's a number of enzymes in this pathway. Ultimately, they will result in this essential cofactor tetrahydrofolate, or vitamin B9. So sulfa drugs actually inhibit the one enzyme in this pathway that's called the FOLP enzyme, also known as DHPS, or dihydropteroate synthase. So the mechanism of action of sulfa drugs is very well understood, as well as the mechanisms of resistance, that is, at least the mutational-based mechanisms of resistance is well understood through structural biology and biochemistry methods. So this is, sorry, let's go back here. Yeah, this just shows the crystal structure of Yersinia pestis DHPS or FOLP enzyme as a representative of this enzyme family. And sulfamethoxazole is bound in the active site, and this is because it is a chemical analog of one of the substrates of DHPS, which is para-aminobenzoic acid. So the plasmid-borne mechanism of sulfa resistance is through acquisition of the so-called SUL genes that encode the SUL enzymes. And these are actually fairly widespread on plasmids, mobile genetic elements, especially the SUL1 gene in particular. According to a recent check of the CAR database at McMaster University, SUL1 is found in approximately 50% of clinical isolates of Acinetobomaniae, for example, and about 45% of Pseudomonas aeruginosa clinical isolates. There are 4 currently known plasmid-borne SUL genes, SUL 1 through 4. And they function through what's called a molecular bypass. They actually create a DHPS enzyme that for all intents and purposes, looks exactly like the FOLP enzyme itself. And we published a paper last year in Nature Communications describing that from an enzymology and structure perspective, they're essentially the same as FOLP, but they do have a key difference in the active site that allows for them to resist the action of sulfa drugs. And this is pretty key that we identify and characterize in our paper. These enzymes have a particular motif that is well positioned in the active site to prevent sulfa drugs from binding there. So they're able then to fulfill the role of the FOLP enzyme so the FOLP itself is inhibited by sulfas, and then these would bypass that drug inhibition to allow progress through the pathway. So inspired from that recent work from last year, I started investigating these couple questions I've delineated here. The first is, what is the origin of the SOL enzymes? As Professor Larson introduced to us yesterday, we're always interested in understanding what is the reservoir of these genes, what are the bacteria that originate or harbor these genes, and actually we don't really know the answer to that. So a little bit of a give you the catch-all to the talk right now, but I have some hints that I'm pursuing. As well, I was also asking the question, are there additional SOL-like enzymes that we just don't know about that could perhaps be new plasmid-borne resistance genes in the future? And for this, I undertook a computational approach using 3 different methods mainly, as well as a fourth that I added on at the end here. So this is based mainly on structure-based similarity methods. So we all have done blast searches based on nucleotide or protein sequences, but with recent developments in AI technologies, especially the AlphaFold algorithm, we're now essentially able to do blast searches with structure. So this was an approach that I used to identify additional members of this family. And this basically leverages the well-known axiom in structural biology and biochemistry is that the 3-dimensional structure of an enzyme is more conserved than the sequence. This allows for more sensitive identification of homologous proteins as well as more sensitivity. I also paired this with sequence-based similarity searches using profile HMMs. And then these 2 searches led to a large collection of tens of thousands of sequences that I ultimately visualized through sequence similarity networks. And I'll walk you through a couple of that network coming up. As well as then using interesting enzymes identified in the SSN, looking at the genomic context to understand, are these chromosomal, are they near mobile genetic elements, do they have any conserved genomic organization? So in terms of the AI-informed structure similarity search, I leveraged 15 crystal structures of the FOLP enzyme as well as our 3 SOL enzymes that we crystallized last year. And I used the fold-seek search algorithm, which essentially allows you to do the blast using 3-dimensional structure. And this algorithm was used to search a variety of databases, including the AlphaFold protein structure database, which has about 200 models of proteins there, as well as the ESM Metagenomic Atlas, which is also in the tens, if not hundreds of millions of models of proteins. As well, the fold-seek search was also used to search the mGenify EBI metagenomics database. So this search resulted in approximately 199,000 sequences. This is potentially including redundant sequences. This was with no filter applied. And actually this data set was quite diverse with, on average, pairwise identity of 22% between protein sequences. And then in terms of the sequence-based similarity search, I trained 2 HMM models using the sequences of SOL1, 2, and 3, as well as a variety of sequences of known FOLP enzymes. And I used the Hammer package to do that. I searched the NCBI and UniProt databases, and then retrained another round of HMMs using now a larger data set from NCBI and UniProt, and then completed a search using the second generation of HMMs. And this ultimately resulted in, respectively, approximately 112,000 sequences for the SOL HMM and approximately 110,000 of FOLPs. I combined those sequences with the fold-seek search results, resulting in about 422,000 sequences. And now I applied a filter to remove redundancy using the CD-HIT algorithm, and this resulted in approximately 54,000 non-redundant protein sequences. Then those 53,000, or approximately 53,000 sequences were visualized using a sequence similarity network. This is pretty ugly, but I'll walk you through it. Basically, every blue point is a protein sequence, and every line refers to relationships between protein sequences where those two protein sequences are related with an e-value of approximately e to the minus 70, so quite related. You can ignore these. These are non-FOLP or sol enzyme, but they would share some sequence or structural similarity. And then I would map the FOLP, which we know is drug sensitive, the 4 plasmid-borne sol enzymes that we know are drug resistant. As well, there have been 2 functional metagenomic surveys of soils that identified about 4 genes that confer sulfonamide resistance. And basically, the upshot of this figure is to show that most of the FOLPs cluster in this large family here, which essentially every bacteria has a FOLP, so that's why this is a very large sort of cluster. And you can see this almost shoulder off the main cluster, which is where Sol 1, 2, and 3 group together. Interestingly, Sol 4, which was the most recently identified plasmid-borne gene, actually packs very closely with the FOLPs, and we're trying to understand why that is from a sequence and structure basis. And next I focused on this category with the Sol 1, 2, and 3 enzymes. And in here there's about 2,000 sequences. And of those, 867 in this cluster actually have either this motif that we identified that was key for resistance, or very similar motifs with maybe 1 or 2 amino acid substitutions. So in other words, they're very highly conserved membership of these sequences in this cluster that have very similar motifs here. And then I looked at the genetic organization of where these genes lie, and found some very interesting trends. So I found a number of bacteria that harbor either these FOLP or putative sulfonamide resistance genes that have this motif very similar to the sol itself, its motif, and they appear to have what could be folate pathway or shikimate pathway genes. So you may think this makes sense because FOLP is involved in the folate pathway, but essentially why is there this kind of divergent FOLP enzyme or gene in this pathway? Which is quite interesting for me. So perhaps there's multiple bacteria out there that have their regular FOLP enzyme and folate pathway genes arranged in an operon, but they have this slightly divergent characteristic in their active site. And these include a couple streptomyces, mostly from Actinomycetes class. I also noticed some interesting trends in terms of synteny. So we know in E. coli K12, the FOLP, again, this is the drug-sensitive, codes for the drug-sensitive enzyme. That is next door to this GLM gene, which is involved in peptidoglycan maintenance. But actually, this genetic organization is conserved in a variety of non-pathogenic environmental bacteria, such as Rhizobialis and Parvibaculums. So again, but these bacteria actually have now, this is the legitimate sol motif that I would predict allows these FOLPs to actually be drug resistant. So I don't really understand what's going on here. There's this conserved synteny, and these are just different pathways or different metabolic processes. So kind of that would be future directions. What is happening with this genetic organization? And then I took a number of these, actually 8 of the sequences that I just talked about, modeled them using the AlphaFold algorithm, and indeed they appear to have an active site that has the very conserved architecture that seems to have this. So the green is actually the sol motif, and the rest of them really resemble that sol enzyme. So indeed, this is consistent with them being drug resistant. And the last slide I'll show of genetic organization is I actually found the Sol 1 gene itself in a variety of actinomycetes that have either multiple AMR genes, but in this case I'm showing as well they have MGEs. So why is it that just soil bacteria have the Sol genes themselves surrounded by a bunch of other ARGs and mobile genetic elements? So that was also an interesting observation. So there's a lot more that I couldn't show for the sake of time. This is a big data set. So for me, what are the implications from this work? I believe the sulfonamide resistome is much larger than thought. We only have 4 of them have actually mobilized onto plasmids, but maybe there's more. We haven't detected them, and maybe in future mobilization events that will occur. And we are planning some experimental validation of the interesting sequences. For this work, it was very essential that we knew what made a resistance enzyme, because from a biochemistry and overall sequence perspective and structure, the FOLP, drug-sensitive versus the drug-resistant sol enzymes essentially look the same, but it's this key motif that differentiated those 2 classes. As well, we observed that a variety of these fall P slot potential cell genes that are co-localized with metabolic genes, and they could be involved in folate pathway or shikimate pathway. So again, are these just intrinsic resistance elements for bacteria that just encoding these putative cell genes next to their normal metabolic processes? And I think this general approach can be applied to any ARG class. Now they're able to essentially blast search with structures, and we have these large databases that are derived from AI algorithms that allow you to do more sensitive, more specific identification of similar proteins. And I think if this, and I know that this is true for specific classes, if not most ARG families, I think the resistance is much larger than we currently know. I've done this analysis with some other families, such as trimethoprim resistance enzymes, as well as some ribosomal methyltransferases. There's a lot more resistance genes, at least predictor resistance genes out there than we think on the surface. And that brings me to the end. I'd like to thank past and present members of our group at U of T, my supervisor, Alexei Savchenko, who's co-localized as well at University of Calgary. Wanted to thank Emily and Nobuhiko at UBC and Simon Fraser, respectively, for help with the SSN approach, as well as members at CFIA and AFC in Canada, especially Michael Frucci for some of our work that was published last year. And thank you very much for your attention.
Hi there. Just a quick question. Oh, I'm Connor Brown. I'm at Virginia Tech in postdoc. I was wondering, I think you had a little bit about it and you hinted at it, but have you observed any coevolution with trimethoprim resistance genes in your analyses? That's all.
Yeah, it's a really good question. I will pursue it further. Off the top of my head, yeah, I did see some co-localization with DFR genes. There's many mobile genetic elements, plasmids, et cetera, that have those co-localized. I can't give you any statistics, how many or particular species, but definitely I did observe that, yeah.
Hi, Peter. Very wonderful talk. Yes, I believe that using your pipeline, we can find or predict more and more new ARGs, right? So AR is very powerful and also can predict many results for us. But how to identify or confirm their real function? Do you have some perspective for the high throughput pipeline to identify their functions? Thanks.
Yeah, no, really good question that hits on a very important point. I think experimentation is not going anywhere, so definitely we have these AI tools and we have these structure prediction and more sensitive searches, but those are trained on the knowledge we already have. So in this case, we already have the knowledge of what motif to look for. For many ARG families, we do have such knowledge, which is great, but for many ARG families, we do not have that knowledge. For example, there are some ribosomal methyltransferases I'm looking at. We really don't understand what are the key active site residues or key sequence characteristics that make it a resistance enzyme and not. I think I look at this approach to guide experimentation, to give candidates. That's what we're doing now, to narrow the list from tens, if not hundreds of thousands of sequences into hundreds, which is viable in terms of being able to express them in E. coli, for example, and verify resistance. So I think this approach is useful for triaging the set to identify candidates for further experimentation.
Okay, thanks very much.
Okay.
Good afternoon, everybody. So my name is Achal Dhariwal, and I'm a postdoctoral fellow at University of Oslo, Norway. And my background is I'm a bioinformatician, and I'm going to talk a bit about a tool that I developed during my PhD, and now I'm working on improving that tool. And the tool, as you can say, is called Resisto Explorer. So in this presentation, I'm going to go a bit on the background, then going to introduce the tool, followed by the workflow and the additional features that I have now been working on to include, followed by conclusion and acknowledgement at the end. So in this conference, we know what's the elephant in the room, AMR, and how many deaths it's causing every year. According to the recent study published in Lancet in 2019, it has been estimated that AMR was responsible for directly causing 1.27 million deaths. And we know it's not only human which are affected by AMR, but it's a global One Health challenge affecting not only humans, but also the environment, as well as the animal and the livestock health. We also are aware that it's a misuse and abuse of antibiotics that we are doing, which has led to actually development of AMR and emergence of antibiotic resistance bacteria. So these antibiotic resistance bacteria have something that now we call as resistome, or the genes that confer or make them resistant to antibiotics. So talking about resistome, it's basically, we all have this microbiota, environmental species or pathogen or commensal bacteria living on us and within us. And it's a collection of all the antibiotic resistant genes that these microbial communities carry is known as resistome. But now the question arises, how do you investigate this resistome? So there are two main approaches. One is called traditional culture-based approach, which still is very useful, but we are aware that many of the bacteria that we harbor cannot be cultured in the laboratory. So this has led to the development of another approach now, which we call as metagenomics, where you don't culture these into the lab. You directly extract the DNA from the sample, then it's followed by sequencing, followed by the characterization of resistant genes. So the whole metagenomic sequencing have now considered as a viable tool for these AMR-based studies. And this has been possible because of the two main features. The first is advancement in next-generation sequencing, and the second is development of bioinformatics tools and databases, which allow us to rapidly identify and characterize ARGs, or antimicrobial resistant genes, in genomes and metagenomes. And what this technology have done, as of now, there's a large-scale studies investigating the resistome or characterizing the resistome, not only in humans, but in the environmental samples and as well as in the animal samples. Just quickly going, how do we do this kind of metagenomic or resistome studies? We start by collecting the sample of interest. This can come from the human environment as well as animals. After getting the samples, you extract the DNA in the laboratory, followed by sequencing. Once you get the sequencing data, the bioinformatics analysis of this big metagenomic data starts. And in this bioinformatics analysis, there are two main steps. Is either you align these reads that you got from the sequencing machine against the publicly available database, for example, CARD and ResFinder, or rather than directly aligning the reads, you can assemble these reads into longer contigs, followed by their annotations. Nevertheless, the output that we got from both of these methods is a matrix, or we can summarize this in a matrix, containing the abundance information of antimicrobial resistant genes across samples. The next step is to explore this data and use various visualization tool or statistical analysis to gain insights from our data. Well, this is not very easy, easier said than done. Why it's not easy? Because this data is big and complex. You need to have bioinformatics skills in order to do that. And even if you have a bioinformatics skills, the issue with the field is there's no one method works best, right? So you have to just explore and explore and explore. And how can you explore this? You use variety of bioinformatics tool. But when I was doing my PhD, I was waiting for some clinical study to get the data and so that I can analyze. But when I was assisting other wet lab or microbiologists in my lab analyzing their data, I realized firstly it's very monotonous to do the same thing again and again, right? And the second thing is like most of the bioinformatics effort at that time, in 2021 I'm talking, was focused on how do you do this upstream analysis where you not annotated these resistant genes. But how to make sense of these annotated resistome abundance profile there was a big gap there. And that's why I developed this tool called Resisto Explorer, which is a user-friendly, web-based, freely available tool for visual, statistical, and exploratory analysis of resistome data. It's publicly available at www.resistoexplorer.no. We published this tool in 2021 in our Genomics and Bioinformatics. You can read if you're interested in knowing more about the features of the tool. To make people understanding about how this tool works, we have this massive online opening course where we have this specific module which enable you to explore and learn how do you analyze this kind of data sets using Resisto Explorer. As of now, Resisto, this is the last month's statistics. It's like it's been used by more than 5,500 users across 71 countries. And since now it's been done, the next question is what's next? I've been graduated last year and then got a postdoc position in the same lab, so I have to do something. So I was like, why not to? And because of this tool, I kind of interact with a lot of users, a lot of microbiologists who have always some bugs in my tool, or this is not working, or how you should do that, you should have these features. And that's why I wanted to integrate all these features in the new version of the tool, which is Resisto Explorer 2.0. So this is how the workflow of the Resisto Explorer 2.0 gonna be look like, because this is under development. So in this tool, we're not gonna start with the downstream analysis, but we actually start from the raw sequencing reads. Then these raw sequencing reads can be quality processed, and you can do the host removal of DNA, followed by annotation of these reads to profile the resistome and the microbial composition. Once you characterize the microbial and resistome composition, you can do all the possible downstream analysis, possible. For example, you can do some basic analysis such as composition profiling, visualizing the composition of the resistome profiles, or you can do basic alpha diversity or beta diversity analysis to characterize the diversity present within the samples and between the samples. You can also do certain advanced analysis, such as differential analysis, which aims to identify the features or antimicrobial resistant genes which are significantly different between the studied condition, can be treated versus non-treated, or antibiotic treatment versus control group. And for this differential analysis, we actually don't have just the standard method that is used for omics data analysis, such as DESAC or EDAR, but we also integrate these, what now called as, and somebody raised the question about this compositional data, the sequencing data. So there are tools such as CODA and LDEX2, which can be used to deal with this kind of data sets. We also do this integrative analysis where we kind of this paired analysis of microbiome profile and resistome profile to answer such questions like whether bacteria phylogeny is shaping the resistome, or which taxa and which ARGs are kind of correlated, or to identify what are the potential microbial hosts of resistant genes. Lastly, we have this functional and network analysis module where we actually utilize the information already present in the public database, such as CARD and ResFinder, to answer some basics but like interesting questions, which I'm gonna present in the next few slides. Looking some of the main features of this tool, now we have this Galaxy-based customized pipeline where we're gonna start with putting the raw data in the software. Then this raw data will be pre-processed. For example, you can do the quality filtering of the reads, trimming of the length reads, you can do the adaptive trimming. And if your metagenome come from the sample which has been like the host of this, the sequence of the host DNA is there, so you can actually remove the DNA. Once you remove the DNA from the reads, you can get the clean reads. These clean reads can be taxonomic profiled using Metaflan 3 or Kraken. And output of this will be, you will have abundance or microbiome abundance table or species count data or species abundance table. Or you can also do the resistome profiling using KMA tool. And you can choose a database of interest because, to be very honest, there's no one database that works best. So you can try different database according to your requirement. And then ultimately you can get the abundance profile. These abundance profile can be directly inputted to the Resisto Explorer 1.0, and then you can do all the downstream analysis that you wanted to do. Yeah. So what we have also do in this Resisto Explorer 2.0 is like, we have designed this tool initially for the case control studies, but we now realize longitudinal studies becoming more and more common because it has certain advantages. But even though it has advantages, there are so many challenges which make this kind of analysis very difficult. But in Resisto Explorer now, we are integrating this tool, which is LDA module, which gives you these methods which are specifically designed for longitudinal data. For example, linear mixed effect model for alpha diversity analysis, which can help you to correct for within sample and between sample variability. And then you can study the effect of actual variable while controlling for the within sample variation. Additionally, some of the visualization methods such as spaghetti plots and horizontal plots, which are designed for longitudinal data sets. Lastly, since the time is short, what we have done is actually we take all the card database and try to answer some of the question, why not to present this card database in a network format. So this is the topological view of the card. As you can see, the pathogen are in the purple, the ARGs are in the orange, and the antibiotics or drugs are in the green, I think. Yeah, the green. Yeah. So this will actually give a topological overview, and you can do various different type of analysis with this. You can do a hypergeometric test to identify which class or which phenotype or which mechanism is enriched in your list of resistant genes. Or we also integrated topological analysis, where you identify the important hubs and nodes, for example, multi-drug resistance bacteria. For example, in this network, this is Klebsiella, which has a lot of connections, and this will be reflected on the topological property also of the network. This is another example where this is a antibiotic resistant profile of Klebsiella pneumonia. These are all the resistant genes in orange and how they confer resistance to different antibiotics. Last example, this fluoroquinolone in the green, fluoroquinolone resistance among different bacteria. The bacteria are in purple. So the time is short, so I just conclude my presentation. So Resist Explorer is a tool that enables researchers to easily explore their resistome data using wide variety of standard as well advanced method. Resist Explorer have an intuitive and interactive web surface, and it can able to analyze your data in real time. Further adding this capability of processing the raw sequencing data, longitudinal data analysis, and advanced visualization will help improving its functionality. What our aim was with this tool was to reduce the gap between data generation and analysis skills and facilitate data understanding, hypothesis generation, and knowledge discovery in the field. The main big objective with Resist Explorer 2.0 is to have this as a one-stop solution for metagenomic-based AMR studies in human, animal, and environmental microbial communities. I would like to acknowledge all my supervisors, funding agencies, as well as EDAR7 and McGill for providing me this opportunity and allow me to present my work. And yes, if you want to try, Resist Explorer is available online, and 2.0 is under testing and development. It will be available shortly. If you have any suggestion, feedback, I'm completely open, just now or even afterwards. I'm willing to get your feedback on this. Thank you so much.
Hello, I'm Dominique Poulin-Laprade from Agriculture and Agri-Food Canada. And I was wondering, can we use your tool, but what we have abundance table? So if all of the heavy lifting has already been done, so starting from the statistical analysis tool. And the second question is, do you have like a version of it that can be local? Like if we have, it's not all data sets that can be put on a web-based.
Yeah, especially because the Resist Explorer when we developed the first version, it was only doing the downstream part. So you can start with the count tables, not from the raw sequencing data. And doing the statistics and analysis visualization is okay for web tools. But now since we have integrated this raw sequencing data module, so we want to have this locally installable version of Resist Explorer. Yeah, so that's definitely in the list. But for the downstream, the first question that you have, yes, you can do the downstream directly going on the web tool because it's not computationally very intensive. The computational intensive step is like doing this raw, processing this raw sequencing data.
I see the present decade as decade of machine learning. We start our day with it, and we carry it with it, and even we end our day with that. But what really excites me is that when this novel paradigm starts interacting with biology. And I'm Vineet. I'm from Virginia Tech, from Dr. Pruden Lab. And I would like to share a few of our findings on ESM2 ARG, a promising deep learning approach for identifying novel antibiotic genes from environmental metagenomes. To start my presentation, I would like to go with a short brief about what are the conventional bioinformatic tools that are used for annotating antibiotic resistant genes. They are BLAST and DIAMOND. These tools work on linear search algorithms. They are really good. I use them in my daily research. How they work is that they take a query sequence, they look in the static database, and they annotate too. They work really good, as I told you earlier. But the problem comes when a novel mutation or if your query sequence is diverging from the database that you're using. And also, the other problem that we have noticed is that, as I mentioned, they work on linear search algorithms. The time for looking into the database increases linearly, and it can cost you a lot of time if your reference database is huge. So here comes a solution for that are machine learning and deep learning models. So what exactly these machine learning and deep learning models are trying to do is that they try to understand the semantic differences between your sequences, and also they'll try to understand the semantics of your sequence and try to annotate them. By this, they can also identify novel ARGs. Application of this machine learning and deep learning started way back. DeepARG, which uses bit score-based similarity and a vanilla neural network to classify your query sequence into one of the 30 antibiotic classes. HMD-ARG uses one-hot encoding and uses convolutional neural networks to classify one of your query sequence into one of the 15 classes of antibiotics. These tools perform good, but they have their own setback. In the present research, we would like to take an advance of research development in machine learning, which are large language models. Usually, large language models are trained on huge corpus of data. Like, to just give an example, ChatGPT-3. Right now, we're using ChatGPT-4. The ChatGPT-3 is trained on 400 billion text sequences or tokens. And major backbone of all these large language models is that you need to develop embeddings. So to give a simple information about embeddings are numerical representation of your sequence data in a high-dimensional view. And in our day-to-day life, we use ChatGPT, Cloud, Gemini, Llama 2. These are basically for English grammar. But when it comes to biological grammar, protein sequence, we have some tools like ProteinBird, AlphaFold, and ESM. In the present research, we have specifically targeted on using ESM2, which was developed by Facebook. And the model that we have used had around 6 layers of transformer encoders and 33 hidden dimensions, and it has around 8 million parameters. The basic background of this ESM2 training is more about masked language modeling. So it will try to mask certain parts of the sequence and try to predict them. So in this way, it is trying to learn the semantics of the sequence. For training our model, we have used existing HMD-ARG dataset, which got published in 2021. And it is curation of 7 publicly available databases, including CARD and ResFinder and few other more. It has around 17K sequences. It not only does ARG classification, but it also has mobility classification and also mechanism classification into 5 classes. And this is our framework. So we have started with taking HMD-ARG. Then we have performed CD-hit40 clustering to split the data into testing and training sequences. Then we have taken the training sequences, trained ESM2, developed embeddings from ESM2, and trained a deep learning model on that, and used it for classification. To make a comparison with the conventionally used tool BLAST, what we have done is that instead of giving a reference database like CARD, we have given the reference database that we got from CD-hit40, that is our trained data set, and our query sequences are test sets. And since we are working with sequential data, it is really important to make sure that your training and test sequences are very divergent from each other, or else it will result in inflated accuracy. So to do this, what we have done is that we have performed CD-hit40 clustering and make sure that our training sequences and test sequences had a max of 40% similarity. And we also made sure that we have covered all the 15 classes in both test and train sequences and try to maintain uniform distribution so that the model is not biased towards any class. And these are our results on the test data set that we have performed. And our model achieved around decently good, around 70% of accuracy. And few classes really showed really good performance, like beta-lactamase and other. And we have also went on comparing with other existing models. Here you can see that ESM2, when we have not stratified data, it showed close to 90% of accuracy, whereas other tools like DeepARG and HMDARG, existing ones, have close to 75, 80, 85, 90 thing. And all the deep learning models performed really very good in comparison with the BLAST. BLAST had like comparably high precision, but low recall. So this indicates that it was unable to identify novel sequences which are not present in its database. Next, we went on validating this on a novel ARG sequences. We specifically mined the literature after 2021 to make sure that these sequences are not present in our training or test data set. And we curated around 20 sequences from different domains, like from duck waste, soil, etc. And this is the performance of the model. The model was actually able to correctly classify 18 out of 20 sequences. There were 2 misclassifications that had happened. But when you see the BLAST was only able to identify 4 out of the 16 sequences. This is because that 4 sequences had a similarity more than 80. I think it was close to 83% and 84%. So yeah. So in conclusion, I would like to say that ESM2 model actually performed really good. But when you are using any large language models like this ESM2, we need to make sure that we are stratifying data properly so that there won't be any bias towards the training or test data set. And also, all the deep learning models really perform good in comparison to BLAST. It seems like the model is performing really good, but there is one big caveat in this. That is, all the models that we are looking now, all the deep learning models or all the large language models, or for per se, all the machine learning models, they're highly biased towards your input data. So if your input data is having one specific class highly distributed, then the models will be kind of inclined to that, and it shows high accuracy for that specific class and less accuracy for the remaining classes. So to specifically improve this thing, we are looking forward to come up with some new embedding techniques, or maybe taking advantage of BLAST and deep learning models together to address this. Yeah, and finally, I would like to thank my advisors, Dr. Pruden and Dr. Zhang, for helping me out of this, and Virginia Tech for funding me, and all the SIPCarians for their support. Yeah, thank you.
Hello, thank you very much for your nice talk. I'm Takuya from National Institute of Infectious Diseases, Japan. And I think it is very promising methodology. And if we find out some novel, newly emerging antimicrobial resistance genes from the environment, do you have any plan to communicate with the public health authorities? Because sometimes it may be needed for the outbreak investigations. So how do you inform the public health authorities to trigger the outbreak response?
Oh, actually, that's a really good question. I think you are looking in the direction of implementing this tool in the surveillance platform so that it can automatically detect if any novel ARG arises. Right now, we are not looking in that direction, but that might be our future direction, to implement this tool in the surveillance program so that it can automatically sense if novel ARG detects. Yeah. Thank you.
Yeah. I think it would be really nice to have lists of these putative genes so that for those of us working in the lab, if we come across something novel, we can verify it so that we can have these verified versus. So we have a lot of great data coming in through these new approaches, but verifying that they're actually encoding for what you think the biological activity is would be really great.
Yeah.
So having those somewhere, even if it's not in the surveillance, would be nice.
Yeah. Thank you.
Hi, I'm Chad Kelly from Virginia Tech. So yeah, I was just wondering, Vineet, so currently it seems like your architecture is mostly focused on just the deep learning by itself, but has there been any consideration to include the card annotation alongside the deep learning as kind of a dual process, if you know what I mean by that?
Yeah, in the future, actually, we are trying to do the same thing to improve our annotations for few specific classes where the model did not perform really good.
Okay, gotcha. Great, thank you.
Then I have one quick question. What you said about the bias of the data, do you think there's any way to correct for that?
Sorry?
You said that there is a bias because of it when you apply your machine learning, because it's biased towards what the main data is putting into your machine learning. So that it's always heavily biased to that. And I was wondering if there's a way that one could correct for that, since you know it beforehand.
Yeah, there are some tools that are already existing to take care of the bias that is happening. Like, for example, if there are few sequences of specific class that are falling down, some statistical tools are being there.
That's what I mean.
But the problem is that if it is only for one or two classes, it can be done. But if it extends on a big level, then you need to think of something else.
Good afternoon, everyone. As was just beautifully pronounced just moments ago, my name is Emma Griffiths. I'm a research associate at Simon Fraser University, where in Vancouver, Canada, where my team develops data standards and ontologies for public health, One Health, and food safety pathogen genomic surveillance. So after all of the wonderful talks that we have had so far right now, but over the course of the conference, it will be absolutely no surprise to anyone when I say that pathogen genomics sequences can be used as molecular fingerprints to help track and control infectious diseases. And furthermore, One Health initiatives can exchange these fingerprints as well as other associated information to understand the prevalence and diversity of different AMR determinants, how they're evolving over time, how they're being transmitted, and how ultimately they impact human and animal health. Now, when we're talking about pathogen genomics data, we are, of course, talking about the sequence data, but we're also talking about the contextual data. And when I say contextual data, what I'm talking about is the sample metadata, as well as the clinical, laboratory, epidemiological, and environmental data, and the methods and provenance information. And all of this contextual information is absolutely critical for interpreting what's going on with the sequence data. And when you don't have good contextual data, you're a little bit limited in what you can do with the sequence data. But when you have good contextual data, the world is your oyster, and you can do all kinds of wonderful things, from doing quality control and comparing results between laboratories, to characterizing different kinds of sequence types. You can resolve outbreaks, you can generate all kinds of hypotheses, and ultimately all of this information can help inform decision-making and to monitor the effects of different kinds of interventions. Now, the thing is that the usability of your contextual data is often impacted by the quality of the information that is initially collected, and also by how you structure that information. Often, contextual data is collected and structured and stored either using organization-specific vocabularies or, more frequently, free text. And these methods can suffer from different kinds of errors. There's often a lot of sector-specific or field-specific jargon. People like to use a lot of abbreviations and shorthand. And all of these issues can contribute to something that we call semantic ambiguity. So this is when the information, the meaning of the information is not clear, both to humans, but also to computers. And this also arises when different labs use the same words to mean different things, or they use different words to mean the same things. And often labs collect and store information using different kinds of data structures. They collect different kinds of information and store it using different concepts, different entities, into different levels of granularity. And ultimately, what this all means is that your contextual information ends up being extremely messy. And it can take days, it can take weeks, it can take longer to clean it up before you're able to integrate it for different kinds of analyses. And it's tricky enough to deal with when you're just talking about private repositories, but the issues get exponentially more complex as the information propagates out to public repositories. So what can we do about this? Before I get into the solutions, I want to introduce to you the GRDI AMR. Now, a number of my colleagues have already done presentations about the GRDI AMR, so some of you may be familiar with it already, but for those who are not, the GRDI AMR is a genomics and research development initiative to support Canada's federal AMR action plan. In a nutshell, I'm about to boil down its beautiful complexity into one sentence. Basically, the goal of the GRDI AMR is to use genomics and harmonize contextual data to understand and identify AMR in the Canadian food supply and also the environment, to understand the risk that those AMR determinants pose, and to potentially identify possible interventions. Now, there are a number of different federal partners that are contributing data and participating in analysis. You can see the list here. They're involved in all the way from public health, animal health, and environmental health. There are also a number of different academic partners, as well as different agricultural and environmental networks. So, when you think about all of the different partners involved in this wonderful initiative, they're all from different sectors. They're using different systems and processes to collect and structure and store information. There are many, many different sample types, different commodities, different environments, different hosts. Everyone's using different sampling strategies. Everyone's using different methods. So you can imagine that the amount and quantity of data is very vast, and it's also very complex. So being able to integrate all of that information is very challenging. So this is where the GRDI metadata workgroup comes in, and it was our task to provide a data standard to enable data harmonization and integration. And after a brief data assessment and a landscape analysis to see what standards already existed, we quickly realized that there was nothing that was really fit for purpose. And so we had to develop something. But that was okay. We didn't have to start from scratch. Our lab at SFU already had developed a modular, interoperable framework for pathogen genomics contextual data. And when I say that it's modular, what I mean is all of the standardized fields and terms are brought together thematically by different categories. And when I say that it's interoperable, this framework was originally developed based on an ISO standard that came out a couple of years ago. The purpose of the standard was to provide guidance for using whole genome sequencing to characterize bacteria in food. When we created this initial framework, we realized that it was actually, you could make it to be pathogen agnostic, application agnostic, and you could use it over and over again simply by adding on extra thematic modules or by enriching or depleting individual modules with new fields and terms. And we know that you can adapt it and customize it for different purposes because we've already done it before. We used this framework to create a data standard that was used to help harmonize all of the different SARS-CoV-2 contextual data during the pandemic. So all of the data coming from the provincial public health labs to the National Microbiology Laboratory, that data standard was used. And we also adapted it for the Mpox epidemic in 2022. You can see how we have adapted it for One Health AMR. All of the thematic modules are on the right-hand side of the slide there. Something you need to know is that we are sourcing all of our controlled vocabulary, all of our standardized fields and terms from ontologies. And I don't have time to give you the hard sell for ontologies today, but basically these are collections of controlled vocabulary that are hierarchical. All of the standardized terms, the meanings of the terms are disambiguated using universal identifiers, and all of the vocabulary are linked together using logical relationships. Now, you don't have to remember that part. What you should remember is that these are open source, and they're developed by different community domain experts. But critically, the vocabulary is meant to be universal in meaning. Okay, and also we tried to integrate as many other existing standards like the fabulous NCBI antibiogram standard. We tried to integrate as many of those as possible. So once we had developed this contextual data standard, our job was actually only half done, because putting standards into practice actually activates a whole slew of different implementation activities. Those kinds of activities include things like consultation, so going back to the data generators, but also the data users to understand whether this data standard was actually fit for purpose. When you are operationalizing a data standard, you can't just give someone an ontology file and expect them to be able to use it. You actually have to build tools and create support materials that teach people how to transform the data that they have in their databases into the standard. And so we have created some tooling that I'll tell you about in just a second, as well as different curation SOPs, reference guides, training material, and so on. Critically, when you're implementing standards, you need to test the heck out of them. You got to make sure that they actually work in real world situations, and you have to have a mechanism for feedback and to be able to integrate requests into the data standard. So you need to have a good development cycle, you need to be version controlling, and basically maintaining a standard. Data standards development is not one and done. As data needs evolve, you have to evolve the data standard as well. And finally, what I just wanted to mention was implementing data standards is actually a big community building effort. There are a lot of different communities of practice that you actually need to consult, in addition to the data generators and the data users. So for example, we have to have a lot of conversations with different semantics or data standards organizations. We have to chat with organizations that are articulating best practices for genomics and bioinformatics, like the Public Health Alliance for Genomic Epidemiology. So there's a lot of work that needs to be done when you're developing and implementing a data standard. Now, I don't have time to tell you all about all of the fabulous solutions to data problems that we have been able to solve using the standard, but I'll briefly just say that we've been better able to establish chains of custody. We can structure more complex sample descriptions a lot better. We can capture nuances of experimental design, as well as different sequencing assays, bioinformatics processes. Using the standard, we can prod people to provide a little bit more information that can help benefit risk assessments, and we're able to create a catalog of other types of information that are not in the standard to better be able to connect folks that are interested in using other kinds of data to interpret the sequence data. So taken together as a whole, the data structure, or the data standard, is helping to improve the clarity and structure of all of the GRDI information for both the human users, but also computers. Just a quick word on the tooling. The data harmonizer is a curation tool that we created back in the pandemic. It's basically a spreadsheet-style text editor application. You download it on your computer. It basically presents a spreadsheet to the user. They can fill in their information using the standardized fields and the pick lists of terms. There's some curation bells and whistles there. It provides validation function, and critically, also automated transformations. So we know that data needs to move. Sometimes data needs to be shared within an organization. Sometimes it needs to be shared within a trusted network. Other times it has to go out to public repositories. And if there's a few things that we know in life, that death, taxes, and the fact that there will never be one data standard to rule them all, those are the top ones. But we also know that all of these downstream destinations for data have their own formats. And we also know that manual restructuring of data is error prone. It also takes a long time and is generally just a pain and is a barrier for data sharing. And so we spend a lot of time working with with collaborators that work at these downstream destinations, whether they're repositories or different initiatives that the GRDI needs to work with. And we spend a lot of time mapping our GRDI standard to their data structures, and we're currently creating interchange formats and encoding that into the data harmonizer, so you can enter the data once in the GRDI format and click a button, and it exports in a lot of different formats. So that you can avoid different, all that manual transformation. So you can see here, I've just popped a few of the different international and Canadian initiatives that we're working with. Okay, so I know that time is up. If you take anything away from this talk, it's that the GRD AMR has created a modular interoperable contextual data standard for One Health AMR. It's being implemented across the federal genomics ecosystem and beyond. And we have also created some tooling and support materials to put that standard into practice. But in addition to the harmonization and the standardization that the GRDI standard affords, it also helps to establish quality frameworks for your contextual data. It helps implement different international best practices. It saves you a lot of time. It really improves the efficiency and basically helps to empower investments contributing to local, national, and global AMR surveillance. And with that, I'd like to thank all of the GRDI partners, all of the collaborators, and of course, all of you for listening. Thank you.
Hi, I'm Clayton from Virginia Tech, PhD student there. And I'm wondering, so you've obviously used this framework for lots of different types of problems for COVID and monkeypox surveillance. How streamlined was it to convert to AMR, which has diverse targets, not just live organisms, but genes, and it's a whole, lots of targets, not just one?
Yeah, so every time we repurpose the standard, there's quite a bit of work involved, actually. I would say that you're probably going to need different standards for different parts of everything that you just said. And there are those ones out there. So we have another standard to deal with the genetic part of AMR. But this standard tackles the phenotypic testing part of AMR, and then, of course, describing samples, isolates, and that sort of thing. So I think we've been working on this for 6 years. And it keeps changing as the needs change. So, for example, we didn't initially have metagenomics as a part of this, but now we do. So, like I said, you're never one and done. So the work continues, but it's very exciting, and it certainly sparks joy in me being able to clean up data. So, thanks.
Awesome. Thank you.
Thank you very much.